Hi, everyone. Thank you so much for being here and joining me for this talk that I'm giving on optimizing the metallothionein system using mineral balancing and the power of hair tissue mineral analysis. My name is Becca Lieb. I am a nutritional therapy practitioner. I'm also an HTMA practitioner, and I am a student in Brendan Vermeyer's year-long course called Functional Mental Health Practitioner. And I specialize in working with individuals who have neuroinflammation, neurodegenerative disorders, and mental health disorders, as well as autoimmunity. What I'm hoping you will gain in this talk is an understanding of what metallothionein proteins are and their purpose in the body. Also, how do we use objective lab testing, whether it be a combination of only HTMA or HTMA blood chemistry, even some urinary markers in order to assess metallothionein dysfunction, assess where there might be bottlenecks in the physiology. And, um, a lot of this is already baked into the brilliance of mineral balancing protocols, but there might be some slight tweaks that you can notice in your clients if you're running additional lab testing. And then strategies that you can implement in order to optimize the metallothionein system, as well as health conditions that are associated with metallothionein dysfunction and why this matters to us. So what are metallothionein proteins? They are specialized proteins that are really essential for copper and zinc homeostasis. They also play a very important role in heavy metal detoxification. And I don't want to spoil something that happens later in the talk, but if you aren't already familiar about metallothionein and how important it is, suffice to say it is very important. Metallothionein is also essential for the optimal functioning of the immune system, and MT proteins even have antimicrobial, anti-candida effects. So you can imagine what might happen in the gut of an individual with metallothionein dysfunction, like what might happen in that gut. And kind of like a chicken and the egg sort of question of what came first, the dysbiosis or the metallothionein dysfunction. Metallothionein proteins are also antioxidants and play an important role in redox homeostasis. One thing that's really cool about metallothionein is that they are present in plants, fungi, and also animals, which is so cool because this is one of these things where we are so interconnected and we have so much in common with other life forms. However, one thing that is unique about human metallothionein proteins is that all human metallothionein proteins contain 20 of the conditionally essential amino acid cysteine. Different MT proteins perform different functions. One example of this is that the parietal cells concentrate a particular MT protein, and that is very important for the synthesis and release of stomach acid. And this is maybe an aha moment because, metallothionein proteins are very rich in zinc, and I think it's kind of commonly known in the functional medicine space that zinc deficiencies lead to hypochlorhydria, and this is actually the mechanism behind that. The metallothionein system is dependent on methylation for a couple of different reasons that we'll go into, but namely, it's interacting and regulated by glutathione homeostasis. We'll go into more, how that is the case. And then also because these proteins are made up of cysteine, cysteine is a conditionally essential amino acid. Under certain circumstances of stress and disease, it is essential, or if you're not eating enough protein that contains cysteine directly. And the transsulfuration pathway, or the cystathionine beta synthase gene, is the gene or the enzyme that transforms methionine into cysteine, which is the substrate that is really important for making these proteins. So I really want this talk to be like connecting the dots aha moment. I want it to be like in The Wizard of Oz, where Toto pulls back the curtains and then suddenly so many things really make sense with more specificity. Because if you're an HTMA practitioner, you know that zinc is super important for heavy metal detox. But do you know the actual biochemical mechanisms behind that? This is what I'm hoping you're going to take away from this talk. We're HTMA practitioners. We know what ionic mimicry is, right? That we know that heavy metals can bind in specific locations in the body and substitute for minerals, and it's dependent on um, the chemical properties of the heavy metals, and do they look similar enough in order to trick the body and substitute in. And when we experience mineral deficiencies, this makes us so much more prone to absorbing heavy metals, which, by the way, is true when you're a gardener and you have soil that's deficient in an essential nutrient. The crops that are grown on that soil are going to be much more likely to take up heavy metals to try to perform their biochemistry to the best of their ability. That's also true for animals, and it's true for humans because we are animals. So this is one example of ionic mimicry, where we can see there, below there is a diagram of a metallothionein protein with 7 zinc atoms bound into this squiggly protein with 20 cysteine amino acids bound. And then above, there is a diagram of the same metallothionein protein, except for what's bound in its place, cadmium. A lot of us will know that like if you're thinking back to X incredible mineral wheel, where it's diagramming a lot of these nutrient interactions, zinc is antagonistic to cadmium, and also vice versa. Cadmium is antagonistic to zinc. And there are other areas in the body where this happens, but this is one of those key areas where this has the potential to happen. And this is actually really intentional and is part of the function of these metallothionein proteins. They're intentionally extremely sticky and they want to grab onto metals. And that is one of their purposes, is to provide a defense mechanism against the inevitable exposure to heavy metals. When you understand that as the function, it makes so much more sense where these proteins are concentrated. So they are concentrated in all these different areas that are diagrammed out on this slide. And actually, if you'd be hard pressed to find an area of the body where there isn't empty. Like, there's metallothionein throughout the body, but it's more concentrated in certain locations. The highest concentration of metallothionein is in the digestive tract. And this makes a ton of sense. So the digestive tract, or the alimentary canal, is technically an epithelial lining, and it's called epithelial, and it has that prefix epi because the alimentary canal is technically external to the body, which is kind of weird to think because it's like going inside of us, but because it's a tube, it's this interface where outside meets inside. And anytime there's a barrier in the body that's compartmentalizing one thing from another thing, it's either an epithelial lining, as in the case of like the digestive tract and other external barriers, or it's an endothelial lining. And, that is a barrier or compartment that's internal to the body. So like a really great example of an endothelial lining are the arterial and vasculature linings. And one of the most famous endothelial linings is the blood-brain barrier. And these linings, they need to be selectively permeable. We are probably more familiar with leaky gut as a disordered process whereby the selective permeability is disordered and things are passing through that should not be passing through, leading to inflammation and the circulation of really noxious and irritating compounds into the rest of the body. Once you understand one structure in the body, it really mirrors and replicates itself in other areas of the body. And endothelial linings have a ton in common with epithelial linings, to the point where they even share a lot of the same tight junction proteins, and they will respond very similarly to the same substances that promote inflammation or promote healing. In a healthy individual, 90% of the heavy metals that we are exposed to, that we ingest, don't pass through the gut barrier, which means, and as a result of the metallothionein system, which means that 10% of heavy metals pass through. Okay, well, with someone who's not healthy and maybe has nutrient deficiencies and has an impaired metallothionein system, it's so all the much more important to reduce environmental exposures because they're going to have a very different physiological impact to the same level of metal exposures. There's going to be more metal that passes through the epithelial lining. Okay, so we talked about gut, blood vessels, and blood-brain barrier already. The lungs are another important epithelial lining, as well as the nasal mucosa, the skin. And then the amniotic sac, including the placenta, also contain high concentrations of metallothionein. And then also when you think about the importance of metallothionein in terms of heavy metal detoxification, it makes a ton of sense that our superstar detox organs, the liver and the kidneys, are also high concentrators of empty proteins because they're doing that hard work of protecting us and clearing out the heavy metals that we're exposed to. Let's just do this thought experiment. Even in a healthy individual, and this would only get worse if someone had an impaired empty system. But so let's say someone's healthy and 90% of the heavy metals that they're exposed to in their diet don't cross over the intestinal lumen. That means that 10% cross over. When that 10% crosses over, they cross into the hepatic portal system where they're taken into the liver, which is also one of these areas of the body that is like a high concentrator of metallothionein proteins. And then of that 10% that crossed into the liver, 10% of that 10% would then cross in from the liver into systemic circulation in the blood. And then 10% of the 10% of the 10% would cross across the endothelial lining of the blood-brain barrier and into the brain. So the total math problem, if you haven't already done the calculation, when you're eating a certain quantity of unavoidable lead or mercury or whatever in the food that we're eating, one thousandth of that amount of heavy metal is ultimately going to end up in the brain, which is completely different from when you're injected with the same quantity of heavy metal. That 100% of that immediately goes into systemic circulation in the bloodstream. And then 10% of that could cross the blood-brain barrier. If you understand the structure and physiology of one epithelial lining or one endothelial lining, you can apply that to all those other linings. They have so much in common. They really have so much in common. To give you some examples, homocysteine can actually break down the tight junction proteins. And that's true in the gut as well as the brain. And this is one of the ways in which impaired methylation can actually lead to leaky gut or to leaky brain and neuroinflammation. All right, pop quiz. True or false, glutathione is the body's antioxidant that protects against the majority of our exposure to heavy metals. Okay, well, hopefully you guys answered correctly because you're in a metallothionein talk. The answer to that is false because actually the metallothionein system is doing the majority of the heavy hitting. But let's go into what's actually going on behind the scenes in the interplay between glutathione and metallothionein. So our body has so many different like thermostats to tell it when to do certain things. And one of these thermostats is the interplay between glutathione and metallothionein. Glutathione is an endogenously produced antioxidant, and it is like a first-line defender when we are encountering environmental toxins. When glutathione is oxidized and turned from an antioxidant in its reduced form into its oxidized form, when glutathione levels are depleted 10%, that's really the threshold that stimulates metallothionein proteins to kick into action and take over in terms of heavy metal detoxification. If we are constantly in a stressful environment, whether it's emotional stress depleting glutathione or environmental toxins like mycotoxins depleting glutathione, or even like heavy metals, that is going to stimulate metallothionein maybe initially, but if this process happens in an uncontrolled way, it's going to like burn out and burn through the resources of the body. So now let's go into like the recipe for optimal metallothionein function. And once I reveal the like how this works, you're gonna be like, wow, this is so simple and makes a lot of sense. It's really not complicated. But when I think about this recipe, I want to think about optimizing the biochemistry and what do we understand about the substrates and cofactors that are necessary in order to make metallothionein and also this interplay with glutathione and how can we extrapolate that to being better clinicians? So firstly, we've got to have glutathione homeostasis in order for this system to work properly. And in order to make glutathione, we need to be running methylation in an optimal way. And we'll go into that more detail. We also have to optimize the transsulfuration pathway. This relates both to the production of glutathione as well as the fact that cysteine is one of these byproducts of the transsulfuration pathway and cystathionine beta synthase. Yeah, we also need to have adequate protein intake in order to have this substrate, this adequate amount of cysteine and, potentially other amino acids for making the MT proteins. We need not only adequate intake, but also digestion and absorption. We need adequate copper and zinc. And we also need to reduce stressors on this system, reducing exposure to heavy metals, other environmental toxins, stress, and then immune insults, like, for example, candidiasis or dysbiosis of other sorts. In order to boost the production of MT proteins, one of the most reliable ways of doing that is to ensure zinc sufficiency. So if we were not superstar HTMA practitioners, we might engage in replacement therapy and take this clinical pearl from the scientific literature and say, oh, okay, I'll just put my client on a zinc supplement. However, we are so much more enlightened than that. we are HTMA practitioners, and we understand that there is a mineral system, and we have to take into account interactions and ratios of minerals and the whole enchilada. And so when I say optimize zinc status, what I really mean is we want to supplement with zinc in an appropriate form, taking into consideration the sodium to potassium ratio, and how incredible that we have supplements like NAK Up or Zinc Matrix Pro, which Valence Nutraceuticals makes. It really doesn't make any sense to use zinc supplementation as a band-aid without also getting at a deeper level and really digging deep in terms of unlearning some destructive lifestyle behaviors and implementing healthy techniques for regulating the nervous system, like the pulling down exercise or meditation or sun exposure or whatever modalities resonate with the client. Okay, so I want to give a little bit of review on the biochemistry behind the methylation pathway and the transsulfuration pathway. So this diagram, it includes the methylation cycle and then also the transsulfuration pathway. The diagram, I actually had to add in some cofactors because it wasn't comprehensive in terms of the cofactors, but a lot of these enzymes, which are encoded by genes and also are potentially susceptible to single nucleotide polymorphisms and genetic variability. But these enzymes are oftentimes metalloenzymes. And this is one of the reasons why it is so easy for there to be a positive feedback loop in the wrong direction when someone is exposed to heavy metals, because methylation and transsulfuration are dependent on metalloenzymes. For example, the MAT enzyme is magnesium dependent, or the CBS enzyme is dependent on iron. Also, the CBS enzyme is inhibited in the presence of excessive unbound copper. A clinical pro about how copper toxicity might impact glutathione production. So to make things like a little bit simpler, I wanted to just list out these nutrients and cofactors. And I'm doing this because it bothers me how often I will see clinicians saying like, oh, my client had impaired methylation. I gave them B vitamins. And like, hopefully, we're HTMA practitioners and hopefully we're not just like sprinkling B vitamin supplements willy-nilly, because we understand that, depending on the client's oxidative rate, if they're in fast ox or slow ox or for lows or something like that, that you could really be pouring gasoline on the fire with the wrong B vitamin supplementation. But with that said, we all have a need for B vitamins in order to run these pathways. And there are other nutrients besides folate and B12. Riboflavin and niacin are also essential within the methylation cycle, and zinc and magnesium. TMG is an awesome methylation support, and it really kickstarts the BHMT enzyme, which is like that shortcut enzyme for recycling homocysteine back into methionine. But what's happening in the methylation cycle is we're feeding this pathway with methionine, which is an essential amino acid. It's very difficult to get enough of this amino acid through a vegetarian or a vegan diet. It really is much more richly found in animal proteins. But so this essential amino acid feeds into this cycle, and then the waste product that comes out, or waste product, is homocysteine. And homocysteine, it is a waste product. It is noxious. It does degrade endothelial and epithelial linings. It creates excitotoxicity. yada yada. Like this is a waste product. However, it's also this incredible substrate and this intermediary that the body then uses through the transsulfuration pathway to make glycine, cysteine, and also taurine, which is really cool. Um, so um yeah, so this is something to keep in mind. And really, the more you understand the biochemistry, the more you can be like, oh, I'm going to pay attention to zinc or magnesium, whether that's on an HTMA or blood chem panel or both. And think about the implications of what that could be doing to this biochemistry. Gut health and improving gut health is integrally a part of a mineral balancing protocol. However, I think that there's a bit of a debate on the extent of what we mean by gut health protocols, because there's a really big difference between a gut health protocol where someone's taking an antibiotic or a prescription antifungal versus, you know, an herbal antimicrobial versus taking a probiotic that crowds out dysbiotic bacteria versus, you know, being on a mineral balancing protocol and supplementing with nutrients that have immune stimulating and antimicrobial properties. Like, for example, you know, the antifungal effect of vitamin D or the antifungal effect of adding in a liver support that's also a biofilm disruptor, that's also indirectly methylation support called NAC. So it's quite clear that optimizing gut health is essential for mineral balancing, but I think that there is some room for debate in terms of what we mean by that. But the reason why I want us to all be in agreement that optimizing gut health is essential for mineral balancing is the following. We need optimal gut health in order to absorb nutrients from food. This is in particular very true for minerals. With most minerals, with a couple of exceptions, we need to have optimal levels of stomach acid in order to cleave those minerals into a form that can then be absorbed. We need an intestinal lumen that is healthy and not super leaky in order to prevent absorption of environmental toxins. Like, we can actually mitigate exposures to environmental toxins by improving the integrity of the intestinal lumen. And our B vitamin status, like when I have a client who comes to me with full-on B vitamin deficiencies, like full-on elevated methylmalonic acid or you know, suspected symptoms of B vitamin deficiencies, the first thing that I think about is their microbiome is probably compromised. And the reason for this is that the majority of our B vitamins are actually made by our microbiome through the fermentation of our food. So improving gut health and improving the diversity and composition of the microbiome will, in some cases, reverse B vitamin deficiencies and actually improve methylation status. And I think of that as like a first line strategy for addressing when a client comes to me with methylation markers that are elevated. You know, we need to be able to actually digest and absorb the protein that we're eating. We need that protein because the protein contains sulfur, which in and of itself is a mineral. And the protein is going to be the substrate of these very important metallothionein proteins. it is going to be the substrate that turns into glutathione. Depending on the composition of our microbiome, this can actually improve what's called the feed efficiency. And the nomenclature for that is because a lot of the research for this comes out of research on livestock and how improving the health of the microbiome can actually allow these microorganisms are capable of recycling ammonia and urea and composing it back into amino acids and contributing in a very significant level to the amino acid pool. And some studies say that, it could be like up to 20% of our amino acids are actually synthesized from our gut microbiome. So there's this potential for and with clinical discretion, because I realize that there is a bit of a debate in terms of what constitutes gut health and gut healing, that doesn't detract from a mineral balancing protocol. But I do think that there is a use case for optimizing the status of the microbiome in a little bit more of a temporary way to create space for accelerated healing, as long as we're adhering to this principle of like doing no harm and not doing things that are counter to the intent of the mineral balancing protocol. We live in the Anthropocene, it's 2025, and all of our microbiomes have fallen from Eden, sadly. There are so many ways in which this microbiome disruption could have happened, but we've all probably had at least one course of antibiotics, and if you haven't, you are so lucky, and I am so jealous. We live in a post-industrial world, and we are exposed to thousands of times the heavy metals and also environmental toxins as our ancestors from a pre-industrial age. Our food supply is contaminated with glyphosate. Glyphosate is actually also patented as an antibiotic, and so we're kind of like low-key consuming glyphosate and other harsh pesticides that have explicit antibiotic or kind of low-key antimicrobial and microbiome disrupting effects. Many of us were born by C-section. I'm one of those. I'm a C-section baby. And, other incredibly sad inventions of the post-industrial age, including hand sanitizer, which is really just like, ugh, what an insane, like cesspool of endocrine disruption and microbiome disruption. And also we live in these like sanitized houses that are very domesticated, and we're not sleeping in the dirt, and we're not, eating a bunch of funky foods, and we're sipping our super controlled coffee that's been tested for mycotoxins, and we're living very, very pure lives that are very different from the lives of our ancestors in terms of the inputs that they were exposed to. So we're not leading, like we are not in an Eden, we're in a fall from Eden. And to what extent, like how do we respond to that? Like what, and I don't have all the answers to this. I don't claim to, but I am thinking about this as a guiding framework as a clinician, in terms of asking this question of what types of interventions do we want to do to maybe mimic some of the inputs that our ancestors got. So whether that, depending on you as a clinician, that might involve, consuming high-dose probiotic therapy. Like, for example, if you haven't heard of this particular strain of Bacillus subtilis, which is a spore-based probiotic called HU58 that Microbiome Labs makes, and can be like very effective for combating bacterial overgrowths in the small intestine or elsewhere in the colon, because it makes antimicrobial antibiotic-like compounds. highly recommend researching that. It's something that I've used in my clinical practice, and it's one of those supplements where my clients um, will take this and be like, oh my God, I cannot believe how much this has had a positive impact on my gut health. Not to say that I think that that is a permanent solution and all we need to do is high dose HU58 in perpetuity, but just something to keep in mind and it might be helpful as a bridge while we're working on these more foundational um, the more foundational HTMA mineral balancing. So maybe purchasing a probiotic like that might resonate with you, maybe doing it a little more punk rock and making your own Dr. Davis SIBO yogurt. There are certain strains of bacteria that are beneficial and that are commensal to the human microbiome that colonize the oral microbiome or the stomach or the small intestine. And these are species of bacteria that have co-evolved with us and our ancestors lived with, and that are extremely susceptible to antibiotic use and are missing in the modern microbiome. But these species, a lot of them are really incredible and produce a lot of digestive enzymes and crowd out these opportunistic and more inflammatory organisms that would otherwise potentially take over in that same terrain. So again, like maybe a little bit more sustainable in the long term to think about like fermenting your own yogurt in this like wholesome way and tending to these microbes, but not going to fix the underlying mineral imbalances and dysfunction that is the result of the heavy metal exposure, which we're going to address with the actual mineral balancing protocol. So as a guiding framework, because I know what I feel comfortable with as a clinician in terms of recommending it to clients, but I don't know what you're going to decide you want to do, and I fully respect what you choose to do with this information. But really just using a framework for making these decisions. Firstly, it is so important as clinicians to operate, even though I'm not a doctor, I'm an NTP, but I believe in doing no harm. And so personally, I am extremely skeptical of any sort of kill protocol, whether it's with a prescription or a more like holistic herbal thing. Extremely skeptical of that and have not, at least within my demographic that I am serving, I've had a lot of negative reactions to those types of protocols. I'm not saying that there is never a time or a place for that, but I think in terms of this principle of do no harm, that's one of the lines in the sand for me. And I know a lot of other HTMA practitioners would agree that those like that an antimicrobial protocol does not marry well with a mineral balancing protocol. They're just doing two different things. Another principle that I think about is that because we live in 2025 in the Anthropocene, even though there are these studies that show that maybe microbes and probiotics have a transient time in the gut, and that even things like spore biotics where they're marketed as like capable of colonizing the gut permanently, there's no such thing as permanent, and that gut health takes active work and active participation. It's not like we can just do a protocol and then go back to the way that we were living before, eating fast food and being super stressed out and, bathing ourselves in Wi-Fi and artificial light, et cetera, and believe that that's not going to have an impact on our health. So it is okay with something like the example that I'm thinking of like taking active work is making your own ferments at home as a way of counteracting living in a really sterile environment. like I like to joke that I've entered my dirtbag microbiome era where like I just want to bring in the filth. Like I think that um our immune systems are trained by what is present in our gut, and we want to be sending that feedback that, you know, we're not living in a sterile environment and we're um gonna outpace the hygiene hypothesis, and we're gonna be like somewhat strategically unhygienic through the use of ferments and yogurts, and whether that's with wild type fermentation or the specific cultivation of particular strains that have been researched. And then also this other principle that metal eliminations are the guiding North Star, that metal eliminations and exposure to heavy metals are one of the things that we know about that actually leads to a durable change in the composition of the microbiome. And from a root cause perspective, this is where we really want to be focusing our priorities and our attention, while also realizing that this might take years to effectuate. And so we got to have a balance of short-term strategies that do no harm, and then also these long-term strategies to optimize the metallothionein system and to eliminate heavy metals. So how do we use an HTMA test to look at the health of the metallothionein system? So first of all, hopefully you understand that the NAK ratio is really telling you a lot about zinc and um, the need for zinc in balance with other nutrients, like, you know, it's perfectly formulated in NAK up. You know, we can look at clues of copper toxicity and how they might be impacting cystathionine beta synthase and the transsulfuration pathway. When we think about copper toxicity, a really classic example is calcium shell. That's kind of like the end game, one of the end games of copper toxicity. And when we think about copper toxicity, it's really also like an end game of heavy metal exposure because of the indirect interaction between copper and zinc. So when zinc is displaced by heavy metals, it creates this imbalance where zinc is not there to counter copper, and then copper accumulates in the tissues as a result. So we also want to be thinking about selenium status and magnesium status and the role that those two nutrients play in methylation and glutathione recycling, iron status as a cofactor for CBS, and yeah, and like, you know, we know that heavy metals could look very falsely low and actually in the, you know, low on in the hair, and then those are the people who are, you know, lacking the vitality to push those metals out, or they could be elevated and show toxicity as well. So like, for example, calcium shell, let's look at this from the perspective of magnesium, right? Because like I already explained how calcium shell is like an end game of copper toxicity and metal exposures. But this, you know, if you were a noob HTMA interpreter, you would say, wow, magnesium is high. No, it is literally, the value of it is high in the hair, but this is not bioavailable magnesium. This is a loss pattern. Calcium affects the permeability of the cell membrane, and we're actually seeing that play out with magnesium where it's not getting in the cell to where it needs to be. So this test needs to be read with nuance, and yeah, so this is actually, you know, an example of how you could see a need for magnesium in addition to, you know, like the other nutrients that would be in this protocol. So we can also pair HTMA with blood chemistry, and I think that corroboration is the name of the game. I love seeing how things line up and are corroborated in the blood work, or potentially seeing nuance and caution where things don't line up. We can look at the methylation markers and corroborate that there is an issue with methylation. So obviously homocysteine, but creatinine is also a great accessible methylation marker because it is produced using a methyltransferase enzyme, so it's a methylation dependent um, end product. Whole blood histamine methyltransferase is responsible for degrading intracellular histamine. And so this can also give you clues about methylation status. Hematocrit, red blood cell, and all of those red blood cell indices like mean corpuscular volume and blah, 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 blah. Like those sorts of anemia markers that are really dependent on DNA methyltransferase and the healthy division of the red blood cells leading to the correct size and proportions and stuff. We also, in the blood chemistry, can get clues about protein sufficiency markers, or even potentially clues about dysbiosis. But so if we have low blood urea nitrogen and low creatinine, or potentially some of these other markers that can go low in the presence of insufficient protein, whether it's a lack of intake or lack of absorption, then that can kind of corroborate the clinical picture through doing just an intake or a chronometer sort of a thing. Then we can also assess glutathione status. Glutathione is a little bit more pricey as a standalone biomarker, and actually GGT through Avexia is like $3 or something, which is just awesome. And I think in a lot of cases is totally sufficient for serving as an inversely correlated proxy marker for glutathione. We can look at serum copper and we can look at plasma zinc, realizing that, what's in the blood is, influenced by inflammation, and they are both acute phase reactants. And also what's happening in one compartment of the body is not necessarily the same as what's happening in another compartment of the body. And just look at the hair as well as the blood for further details. And when another marker of assessing copper dysregulation and copper toxicity is looking at a ratio of copper to ceruloplasmin, which is actually what's done clinically in more extreme cases of this, the genetic disease, Wilson's disease, where you have a lot of unbound copper and a genetic inborn, a defect in the ability to produce ceruloplasmin effectively. You can also, look at the iron panel, correlate that to CBS. You can look at the transport proteins as clues of are you getting exposed to heavy metals? Because these transport proteins like transferrin and ferritin and albumin, in addition to being an iron binding protein or a zinc binding protein, they also bind to heavy metals. And so you can, and they also respond to oxidative stress and inflammation. And so a lot of the time, for example, ferritin will elevate in the presence of heavy metals. It's a positive acute phase reactant. But what's one of the reasons why there's a storage metal protein, usually for iron, but can bind to other heavy metals, it's getting elevated because you're getting exposed to heavy metals. It's not necessarily an elevated ferritin isn't necessarily only an iron dysregulation issue. It's potentially a heavy metal exposure issue. And the way that you would kind of distinguish between the two is you'd look at, okay, well, is ferritin elevated, but then what does the iron panel look like? And then you can look at the copper to zinc ratio, which is a really excellent kind of broad spectrum, very, very clinically established and well-researched marker of overall inflammation. Yeah, and then you can screen, there aren't, you can screen for B vitamin deficiencies, but there aren't really the best ways of measuring those markers because you can get false low or falsely high levels of those. However, there is one marker, like if you really are concerned about B12 deficiency, methylmalonic acid is like the gold standard for seeing if a B12 deficiency is present. It's a little bit of a pricey standalone marker, but if there's an unlimited budget, then maybe something to consider, or if there are particular symptoms that are lining up more with B12 deficiency, then something to consider just to rule out. There is a urinary marker called cryptopyrrole, which is very genetically driven. Like certain people will have more of a genetic predisposition to elevations in cryptopyrrole. And this is it's kind of like poorly understood, still being researched marker, but what we do know about it is that it tends to like lead to zinc deficiency. And when zinc status is corrected, then the cryptopyrrole will go back into a normal range. And this is a commercially available lab through DHA laboratories, through a urinary sample. And then, if you happen to also be running an organic acids test, which I actually really love and prefer to using stool sample testing to test for the status of the microbiome, then you can infer things through that. Like, for example, we know that metallothionein proteins are very effective antifungal agents. And so if someone has an oat with a lot of elevated fungal markers, then we would suspect that they probably have issues with their metallothionein system, in addition to all of the leaky gut and like other issues that come along with candidiasis or fungal overgrowth. Okay, so how can we be strategic in our supplementation? The vast majority of everything that I'm going to talk about is like already taken into account with the way that HTMA protocols are built. But I do think that we want to be actively reviewing the lab work nonetheless, and identify, okay, where are there bottlenecks in the biochemistry? So one really great example of this is, how do we decide how to support methylation? How do we support methylation? If my starting point with supporting methylation is supporting the gut and supporting minerals, because it's so non-negotiable. And then if that isn't enough to normalize the physiology, then I'll consider adding in some of these other therapeutics. So maybe, a B vitamin. It's probably already in the mineral balancing protocol with the appropriate doses for that client's oxidative rate. Maybe a little bit of TMG. Really great for the liver, really great for supporting methylation. Maybe a little bit of phosphatidylcholine, already baked into the mineral balancing protocol, like really helpful for not only supporting bile and supporting acetylcholine and the vagus nerve, but also, phosphatidylcholine is this like super methyl donor where it actually has 3 methyl groups that are bound to it. Creatine, taurine, glycine, and acetylcysteine you could think about as methylation supports. Roughly half of our body's methylation demands are just through the demand to produce creatine. And one thing that I would see in a client's blood labs that would make me want to supplement with creatine is if in their CMP they have low creatinine, which is the metabolic waste product of creatine. So that would be like a slam dunk. Taurine, this is also methylation support. It's also a cofactor for bioconjugation. And it is also, a byproduct of the transsulfuration pathway. And taurine is built into certain types of mineral balancing protocols. It's very helpful for calming people down, especially in 4 lows. And so this is something that is already kind of like baked into the mineral balancing protocol. Glycine, similar to taurine, is, one of these conditionally essential amino acids that's made from methionine via CBS. And glycine is also a neurotransmitter. It tends to have a calming effect. NAC, excellent for the liver, excellent for its antifungal properties, its biofilm disrupting properties. So there are certain considerations where I might place more of an emphasis on NAC. And I really think that it's great to have this menu of different options because it is so often where, a client might, like maybe they should be on a certain type of methylation supplement, like for example, TMG. They should be on TMG. And maybe it's leading to a Herx reaction. In the long run, they would benefit from it. Or maybe they really are having an adverse reaction to it and it's making them really irritable or manic. As is the case with my clientele where that would be, a big concern and we'd want to pull back. Then I think it's so helpful to have this menu of options because you don't get boxed into one type of methylation support. And you can use other ones that are gonna be more gentle on someone who's maybe more sensitive and reactive to certain types of methylation support. And also, while a lot of these are baked into the protocol already, there is room for nuance of something like creatine or, these other supports where you can corroborate with the blood work and see like, okay, we can make this more targeted. And why does this matter? So there are a lot of health conditions that are associated with metallothionein impairment. One of my favorites to consider is hypochlorhydria, which is when there's insufficient stomach acid, because it sets the stage for so much of the other dysfunction in the body. But we actually need the production and secretion of stomach acid is regulated by metallothionein. This is the reason why a clinical pearl that a lot of us are already aware of is that we need zinc, adequate zinc, in order to make stomach acid. This is the reason why. So, like what we're doing as practitioners is creating a potential off-ramp for individuals who've been on supplemental stomach acid for years. And we have the potential to actually reverse that by optimizing the metallothionein system. Not something that's going to happen overnight necessarily, but this is something that we really have the ability to do. And, a lot of different neurodegenerative, neuroinflammatory disorders are also really tied to dysfunction in the system, as well as cancer. So definitely something that we want to optimize in the long term. And in conclusion, we can go into this biochemistry and attempt to understand all of these mechanisms as much as possible. That is a noble feat. It's going to make us better practitioners. But at the end of the day, the human body is infinitely complex, and we don't understand everything about the mechanisms. We don't understand everything about the interplay. One of the beauties of the HTMA test is how nonlinear it is, and it kind of gives this glimpse into the domino and ripple effects that happen that are really beyond the realm of human comprehension. And so to practice like a bit of humility in this, and also we can recognize that we don't have to understand literally everything that we're doing in order to help people, I think is so important to hold on to. We are always learning and discovering new truths about the body, and the understanding of the biochemistry, the understanding of the mineral interactions is giving us a framework for being better clinicians, but it isn't a substitute for our own clinical judgment and intuition. And there's always that X factor for healing. So thank you so much for listening. I hope that this was helpful. And here are some citations if you want some further reading.